A very good morning, students. Welcome to science session. And now here in this session we are going to see a new topic, that is unit 18, heredity. Now in this lesson we are going to see what is chromosome, what do you mean by gene and DNA, and the very interesting Mendelian law distinguished between the phenotype and the genotype, what is monohybrid cross, dihybrid cross, structure of chromosomes, classification of chromosomes based on the position of the centromere, replication of DNA, classification of chromosomes and gene mutation. And we are going to learn about a new chromosomal abnormality which is called as the Down syndrome. Right, now let's go into the session, children. We know very well that the living beings produce offsprings of their own kind, isn't it? Children look similar to their parents. Sometimes they look more similar to their grandparents. Color of the eye, color of the hair, shape of the nose look similar to their parents or the grandparents. Now, this is because they inherit the traits and characteristics from their parents or from their grandparents. Have you ever wondered why do we inherit traits and characteristics from our parents or from our grandparents? Sometimes we may be very close to that of the grandparents, that is, we may inherit some of the characteristics very close to our grandparents, isn't it? How do we inherit characters from one generation to the other? This is because of the genes that we inherit from our parents, right? So these genes are responsible for the physical outlook, for the physical appearance, and also for the biological functions that takes place inside our body. So there's a branch of science, or you can say the branch of biology, that deals with what is genes, what is genetic variations, what do you mean by heredity of the living organisms. So all these are called as genetics, right? Now here we are going to learn about what is heredity, chromosome, DNA, genes, and the replication of the DNA. How do the characteristics get transmitted from one generation to the other generation? So first terminology that we are going to learn today is the heredity. Heredity is nothing but the transmission of the character from one generation to the other generation. That is, transmission of the gene from one generation to the other generation. So all these will happen only due to the chromosomes, right? So first we are going to learn what is chromosomes, what it is composed of. Now how is the genetic material being transmitted from one generation to the other generation? So first we are going to learn about who is the father of genetics and what is experiment is about. So the father of genetics is the Gregor John Mendel. Now look into the picture, children. He is the father of genetics, Gregor John Mendel. He was an Austrian monk. He discovered the basic principle of heredity through his experiment. So what is this experiment is about? So in his leisure time, he started his famous experiment on the garden pea plant. You know what is pea plant, isn't it? Right? Now he conducted his experiment for about 9 years from 1856 to 1865, and he had been working nearly or you can say he has dealt with 10,000 pea plant of 34 different characteristics. Now what do you mean by this characteristics? Now he has chosen 7 pairs of contrasting characters for a study. Now what do you mean by contrasting characters? Now let's see that. Now these are the 7 pairs of contrasting characters of the pea plant that was used by Mendel. The first one is the seed shape. seed color, seed coat color, pod shape, pod color, flower position, and the stem length. Now these are the dominant character and these are the recessive character. Now what do you mean by dominant character? The character which express itself, which get dominant, as the name indicates, it gets dominant, right? And the recessive character is one which get masked, which gets suppressed. These are the two different contrasting characters of the pea plant that was used by Mendel. Now here the dominant character is the round, perfect round seed, and the wrinkle seed is the recessive character. Yellow and green. So green will be the recessive character and yellow seed will be a dominant character. Now seed coat, if you see, it will be in colored one, and here it is white in color. Pod shape, that is the outer cover of the seed, will be an inflant one, and here you can see the construction over the pod shape, right? And here pod color will be green, that is the dominant character, and yellow will be the recessive character. Here you can see the auxiliary flower positions, right? Apart from the terminal is only at the top of the plant, right? Only at the tip you can see the flowers, but auxiliary is at the auxiliary nodes, you can see. And the stem length, here you can see a long stem, and here on the other hand it is a short one, right? So that is the recessive character. So these are the 7 pairs of contrasting colors that was chosen by Mendel. Now what is the reason why it is chosen pea plant? Why pea plant is chosen? So let's see that it is because he has chosen the pea plant because it has a lot of advantages when he is doing his experiment. That is, it is very advantageous for his experimental work in many aspects, you can say. So first one is that it is naturally self-pollinating, right? So it do not require any agents for its pollination, and it is very easy to raise the pure breeding since the self-pollination takes place, that is within the flower. Now the next one is that a short lifespan is seen in the pea plant, so it is possible to follow several generations. So within a short period you can see several generations, and it is also easy for cross pollination also. So the cross pollination can also be taken very easily, and it has a well defined contrasting characters, if you see, and the flowers are bisexual. So these are the reasons why he has chosen the pea plant and become very successful in his experiment. So first we are going to see about the crossing, that is monohybrid cross. That is the inheritance of one gene from one generation to the other. We are going to take only one pair of the contrasting character, and we are going to do experiment and show how this one particular character or the contrasting character is being it's been transmitted from one generation to the other generation. Now here you can see the monohybrid cross, that is the inheritance of the one gene, right? So the crossing involving the inheritance of only one contrasting character is called as the monohybrid cross. Now what is that single trait that is being crossed here? Is the tallness and the dwarfness. That is, these are the two contrasting characters, isn't it? So now here the tall plant is represented by T, capital T and capital T, and the short plant or the dwarf plant is denoted by small t and small t. Now if you see the parent generation is denoted by T, and T for tall and small t for the dwarf plant, right? So this pure breeding tall plant and pure breeding dwarf plant is the parent generation, right? Now here this capital T is nothing but the dominant condition. Now what do you mean by dominant condition? Now when you see two factors of two different characters, when they are brought together, one character that express itself, the dominant itself is called as the dominant character, and the character which is getting masked, which is getting suppressed, is called as the recessive condition. So it is represented by a capital T, that is the dominant character, and a small t is the recessive condition. Now if you see the two factors when it is making and contrasting characters, when they are brought together, these are called as the alleles, or it is called as the allelomorphs, right? And each member of this pair is contributed by one parent, right? So this is contributed by one parent. So this is about the alleles and the dominant character. Next we'll see what is the homozygous and heterozygous. Now when the factors occurs, when the inheritance occurs, there may be an alike or the unlike pairing can take place. Now pure breeding, that is if it is in capital T and capital T, so this is the pure breeding, isn't it? So when they are alike, right, so they are called as the homozygous, right? So here you have both the dominant character, they may be alike, isn't it? So these are the dominant character that are brought together, so it is the homozygous. Even if the recessive character are brought together, then it is also called as the homozygous, but it is in dwarf homozygous, and this is the tall homozygous, because both the dominant character are brought together, and here the dwarf, that is the recessive character, is brought together. Now what is heterozygous? Heterozygous is when they are unlike, right? So one dominant character and one recessive character, when they are brought together, so it is called as the heterozygous. So this is a combination of a dominant and a recessive character. Now let's see the generation. Now we said that this is the parent generation, where it is represented by capital T for the tall plant, and small t for the dwarf plant. Now if you see the gametes, it is represented by capital T and small t, right? So when the selfing, that is the self crossing, takes place in the first generation, if you see, actually all the plants are tall, right? And if you see the phenotypic ratio as well as the genotypic ratio, all the plants are tall, because the dominant is expressed here in the first generation. Now what is genotypic and phenotype? So the internal expression, that is the genetic expression of an organism, is called as the genotype. So that is the genotypic ratio, the ratio that is given in terms of the genetic expression of an organ is called as the genotype. Now phenotypic ratio is the external expression of a particular trait, so that is called as the phenotypic ratio, right? So this will give you the appearance, external appearance, whether it is tall, whether it is the short, and that ratio is given as the phenotype. So all the plants are tall in the first generation. Now if you see the second generation, the gamete is represented by small t and capital T, right? So when you see the combination, capital T together, small t and capital T combination, small t and capital T combination, and small t combination, right? So this is about the second generation crossing, right? Now if you see in this, so if you see the tall homozygous, here this is the tall homozygous, isn't it? So both the dominant character are together. And if you see the dwarf dominancy or the dwarf homozygous, so this is about the dwarf homozygous, where the both the recessive character are combined here. Now this gives you the heterozygous, tall heterozygous, where the ratio is 2. Here you have 2, isn't it? So 2 combination, so twice this combination occurs, isn't it? The recessive character and the dominant character together, right? So 3 different types are obtained in the second generation. So first one is the, what is that 3 different types? First is the capital T together, small t together, and capital T and small t together, right? So what will be the ratio of the genotypic? So 1 is to 2 is to 1, right? So we got 1 capital T together, one small t together and 2 times we got small t and capital T together, isn't it? So this is called as the genotypic ratio, where the genetic expression of an organism is represented in terms of a ratio. Now if you see the phenotypic ratio, the overall external expression of the plant, now here you can see that all the 3 plants are tall and one is the dwarf plant, right? You see the ratio between the tall and the dwarf plant. It is 3 plants are tall and one is the dwarf plant, right? So the ratio will be the 3:1, and this gives you the external expression of an organism. So this is the phenotypic ratio. So this is about the genotypic and phenotypic ratio. So in the monohybrid cross, in the first generation, all the plants are tall, and in the second generation, 3 plants are tall and one is a dwarf plant. So the genotypic ratio is 1:2:1 and the phenotypic ratio is 3:1. Now let's move to dihybrid cross. Di means 2, right? The inheritance of 2 genes, right? So now the dihybrid cross involving the inheritance of 2 pairs of contrasting characteristics at the same time, right? Now what is the 2 contrasting characteristic that is chosen? The shape and the color of the seed, right? That is round and wrinkled seeds. And if you see the color of the seed, it is the yellow and green color, right? So these are the 2 contrasting characteristics that is chosen by Mendel. So Mendel crossed pea plants having round yellow seeds with the pea plant having wrinkled green seeds, right? So capital R represents the round seeds and small r represents the wrinkled seeds. Capital Y represents the yellow seeds and the small y represents the green seeds. Now here you can see the hybrid crossing. Now this is the parent generation. The gametes of the parent generations are represented in single, that is RY round and yellow, and small ry, that is wrinkled and green seeds, right? So he first crossed the pure breeding pea plant having the yellow round seeds with the pure breeding pea plant having the wrinkled yellow seeds, and he found that only round yellow seeds were produced in the first generation. So this is F1 generation. So in the F1 generation, only the round yellow seeds are produced. So from this he is concluded that the round shape and the yellow color of the seeds are the dominant character, that is R and capital Y are the dominant traits and small r and the small y are the recessive traits, right? So when the hybrids of the first generation having the round yellow seeds were cross bred by the self pollination, you get the second generation. Now in the second generation, now here you can see it is represented in the form of a checkerboard, isn't it? So this is called as the Punnett square. Now what is Punnett square? Punnett square is nothing but the checkerboard. Now you know what is a checkerboard, isn't it? So it looks like a checkerboard, right? So this is devised by a British genetist whose name is R.C. Punnett. So it is named as the Punnett checker or the Punnett square. So this gives you the represents, this checkerboard represents the probability of all possible genotypes of the offsprings in the genetic cross. Now here you can see all the possibilities of these combinations of the gametes in this second generation, isn't it? So since it gives you the probability of all possible genotypes, it is called as the Punnett square or the checkerboard, right? Now here in the F2 generation, now there are round yellow seeds, round green seeds, wrinkled yellow seeds, and wrinkled green seeds. Now here you can see the ratio of the each phenotypic, that is appearance, that is external expression of the seeds in the second generation. And if you see the combinations, here you can see 9 yellow round seeds, 3 yellow wrinkle seeds, 3 green round seeds, and 1 green wrinkle seed. So what will be the ratio? The ratio will be 9:3:3:1. So this is nothing but the F2 generation phenotypic ratio, that is the external appearance, right? So it is called as the dihybrid ratio. This is also known as dihybrid ratio, where the two different contrasting genes are inherited and the possibilities are given in the form of a checkerboard. Now it is also concluded that the each characteristics or the trait remains independent and maintain their identity in the gametes, right, when it is inherited, right? Now if you see the result of the dihybrid cross, 4 types of plant are found. That is, a dihybrid cross produces 4 types of F2 offsprings. Now here you can see 4 types, 1, 2, 3, 4 types, isn't it? 4 types of offsprings in the ratio 9:3:1. And if you see 2 new combination traits with round green and wrinkled yellow. So these are the new combinations from the gametes, isn't it? It also appeared in the dihybrid cross, especially in the second generation, right? These are the results of the dihybrid cross. And if you see, based on these monohybrid and the dihybrid experiments, Mendel proposed 3 important laws, which is called as the Mendel's law. Now what is this Mendel's law? The first one is the law of dominance. The second one is law of segregation or the law of purity of the gametes. And the third one is law of independent assortment. Now let's see them one by one. Now based on the monohybrid and the dihybrid cross, Mendel proposed 3 laws, I said, and these 3 laws together is called as the law of heredity. The first one we said is the law of dominance. Now what is this states that? Now this law states that when 2 homozygous individuals with 1 or more sets of the contrasting characters are crossed, the characters that appeared in the first hybrid are dominant, and those that do not appear in the F1 are the recessive characters. Now what does it mean? Now when 2 homozygous individual with more character or 1 set of contrasting character. Now here you can see 1 set of contrasting character, isn't it? The purple and the white flower, right? So this 1 set of contrasting characters, when they are crossed, the one which appears in the first generation. Now here the purple appears in the first generation, isn't it? So this will be the dominant character, and the one which do not appear, now which do not appear here, the white do not appear, isn't it? So this will be the recessive character, right? So the one which do not appear in the first generation will be the recessive character, and which appears in the first generation is the dominance character. So this is called as the law of dominance. Now here you can see that the parent gamete, right, of the purple and the white flower are crossed, and in the first generation you can see a purple flower, isn't it? So the purple flower will be the dominant character and the white flower will be the recessive character. So this is about law of dominance. Now the next one is the law of segregation or law of purity of gametes. Now this law states that when a pair of the contrasting factors or genes or the allelomorphs are brought together in an heterozygous or hybrid, the two members of the allelic pair remain together without mixing, and when gametes are formed, the two separate out so that only one enters into the each gamete, right? Now these are the two characteristics, isn't it? The contrasting characters. This forms the allelomorphs, isn't it? So they remain together without mixing. So without mixing, they are together, right? And when they enter into the gametes, so these are the possibilities that you get, right? So they separate out, right? So when they enter into this pair of chromosomes, which consist of the genes, they enter into the gametes separately. They separate out and they enter into it, so that each enters into each gamete, and you will get the different possibilities, right? So the two copies of each gene separate out and they will end up in different gametes, right? So this is called as law of segregation. The next one is the law of independent assortment. Now, this law states that in case of inheritance of two or more pairs of characters simultaneously, the factors or the genes of one pair are sought out independently of the other pair. Now, we saw an example of the round yellow seed and the wrinkled green seed, isn't it? Now, we'll see this once again. Now, with this example, we can understand this law or explain this law. Now here we said that these are the parent generation, right? Now this parent generations in the first generation gives you the most dominant character, and here we said that the dominant character is nothing but the round yellow seeds. Now here you can see the dominant character that appears in the first generation. This is the phenotype. The appearance will be a round yellow seeds, right? Now what will be the recessive character? The recessive character will be the wrinkle shaped and the green color seed, isn't it? Now here these two are crossed together. That is, the pea plant having the round yellow seeds were crossed with the wrinkled green color seeds in the second generation, isn't it? And we got a combination, isn't it? Now here we got a combination in such a way that according to this law, the segregation, that is the segregation takes place, isn't it? So the segregation of 50% of the round seeds and 50% of the wrinkle seeds is independent. From where? From 50% of yellow and 50% of Y, isn't it? That is the green seeds. Now here this is nothing but the segregation from one pair of contrasting character. Now here we saw that two pairs of contrasting character, isn't it? The first one is the round and the wrinkle seed. So this is one pair of contrasting character. What is the other pair of contrasting character? Yellow and the green seeds. So we have two sets of the contrasting character. So the segregation takes place independently, right? So segregation of 50% of R and 50% of the small r is independent from the 50% of the yellow and 50% of the green seeds, isn't it? Now if you see 50% of R as Y, that is yellow, and 50% as small y. And thus total, how many gametes we got? We got 4 genotypic gametes are formed, isn't it, or produced. This law of independent assortment, that is, the genes of a pair of the contrasting character are sought out independently, segregates out independently of the other pair. So totally we get in this the 4 different combinations are obtained, that is round yellow, round green, wrinkle yellow and wrinkle green. So these are the law of heredity. Now, so far we have seen about the monohybrid cross and the dihybrid cross, isn't it? So we'll continue with the lesson in the next class. In the next class, we'll be dealing with chromosomes, DNA, gene, types of chromosome based on this position of the centromere and based on the function, replication of DNA and mutation. With this, we'll complete this lesson. So we'll continue the remaining part of the lesson in the next session. Now your homework for today is that in page number 272 you have long answer questions. In the first question is explain with an example the inheritance of dihybrid cross and how it is different from the monohybrid cross. I have uploaded the PDF. You can refer the PDF and you can get the answer for this question in the PDF. Please do write this question with the diagram in the classwork and submit the classwork in the school office on Friday. We'll meet you in the next session, children. Thank you.